Hello guys, welcome back to my channel Anamika's World of Science. So today we are going to discuss about animal diversity or the animal kingdom. So if you're watching this YouTube channel for the first time, as usual, I'm saying that do like, share and subscribe to my channel and press the bell icon so that you don't miss another update from Anamika's World of Science. So talking about this video can be used as a reference video for the plus 1 students, plus 2 students, entrance students as well as degree first year students whosoever are doing the core paper on zoology, a complementary zoology portion. You can use this video as a reference here. So let's go to the lecture.
So let's go to the discussion. So in this video we'll be talking about the classifications of animal kingdom. So you can see a lady there. She is Jane Goodall. She's a primatologist and this is one of her famous quote. So there's a quote as well. I think I'd like to be remembered as someone who really helped people to have a little humanity and realize that we are part of the animal kingdom, not separate from them. As we all know today, we are thinking that we are somebody very unique than or the animals. But we should remember that we are also a part of this animal kingdom, including the monkeys, then the dogs, cats, elephants, lions. We are all part of their animal kingdom. Don't forget that we are a part of this animal kingdom. This quote will give you that remembrance that you should not forget who you are. So let's go to the next slide. So I'll be telling you that usually the animals are being classified in 4 fundamental features. They are the arrangement of the cell, that may be the body symmetry, the nature of xylomes, the pattern of digestive, circulatory and reproductive system. So the level of classification, I'll be talking the first level of classification, that all animals or all members of an animalia doesn't show the same pattern of organization. Level of organism can be different in case of animalia. For example, sponges are low cell aggregations. So those sponges are really just cells in cluster. So they are called the cellular level of organism. And in case of coelentrates, the arrangement of the complex is usually an example of tissue level. That is, the cells are being aggregated into tissues and they perform the same function. There is a classification of organism. And in case of platyhelminthes, the tissues combine to form organ. So they will be doing the same activity and an organ will do the same activity together. In case of animals like annelids and the above phyla, the organisms are associated as a functional system. That's called the organ system level. So just take that we are animalia group. In case of us, we have organ system level. You know the digestive system, you know the circulatory system, we had an excretory system. So these all the system consist of different organs combined together to form a single system for doing a single type of work. In case of excretory system, that full system is used for the excretory purpose, for throwing out the excretory products from our body. In case of digestive system, the digestive process is for getting apt energy for us. Likewise, there are many different types of classifications. So you will detailedly understand it in the upcoming slides. So you can see a slide had put some pictures: cellular level, tissue level, organ system level and an organism. We know what is a cell. So inside a cell there are many molecules which are combined to form a cell. These cells are aggregated. These cells have their own unique works to do. when clusters of cells which are doing same kind of work join together, they become a tissue. And when some tissues which having same identical work will combine to form an organ level. And organs which are doing almost same type of work, they will be combined to form an organ system level. Finally, these organ system levels are all combined together to form an organism. That's all. It's very simple. Zoology or this studying about the animal kingdom is very easy. And there is another way of classification of circulatory system. The circulatory systems are of two types. First one is an open circulatory system and the second one is a closed circulatory system. You know cockroaches. Ever open the cockroach and see what is inside it? No, right? So inside a cockroach there is a heart. It's not a heart, but it is a, it's a type of a fake heart, we can say. There's a heart and each heart will impulse out homolymph and it will be covering all around the body. They doesn't have any veins, they don't have an artery to pump the blood through. Each and every part of the tissue is being filled with blood. I will show some examples of photos so that you will understand it better. And in case of a closed circulatory system, we can take humans. We have arteries, we have veins, we have a heart which will pump the blood, and our tissues are not soaked in blood because each veins and each arteries, the blood are flowing inside the veins and arteries. We can simply say that if an organism have a perfect path for pumping a blood, they can be called as a closed circulatory system. And in another way, if an organism doesn't have veins or arteries or any other blood vessels to carry the blood out, and such organisms can be classified into open circulatory system. Now there is a new classification on the basis of symmetry. Have you heard the word symmetry? Do you know what is a symmetry? It is a quality of being made up of an exactly similar parts facing each other or around an axis. Or we can cut an organism into two equal halves. Such type of is being called symmetry. So in case of an organism, for example, you can see there a sponge. A sponge, if you are cutting around the plane through the center of the axis, you cannot get two equal halves. So such an organism in which if a plane passes through the center of the axis and does not divide them into two equal halves, then they are called asymmetric. And if we are taking another thing like starfish, or you may see them, it will be like a star, right? But we can pass through any axis so that we will get two equal halves. So when a plane that passes through the center axis dividing the organism into two equal halves, that's what we call a radial symmetry. In case of a bilateral symmetry, just take our human body. We can only get equal halves when we are passing through the center, from the head to the nose, then to the mouth. Likewise, such a cut will give two equal halves. Other than that, if we cut the head out, you will never get an equal half. If we cut through the stomach region, you won't get an equal half, right? So that's what we call a symmetry. You can see there. Some case of third classification, that is diploblastic and triploblastic. I know this is a new word for the +1 students, so kindly listen. Usual organism has three layers, like an outer ectoderm, middle mesoderm and inner endoderm. This condition is called triploblastic. If an organism have all these three, like an organism can be called triploblastic. Just look the picture below. The right side picture, you can see outer circle, you can see the ectoderm, You can see the endoderm in the center. You can see the endoderm, and in the middle, that yellow color, that's what we call the mesoderm. And now just took the first picture. What does that mean? In case of that, the bilaterally symmetrical animals are triploblastic. Bilaterally symmetrical, you know, if you cut the animal into two equal halves, just passing through the center, you will get an equal half. So such animals have triploblastic condition. That is, all these three together. They produce three germ layers that are ectoderm, endoderm and mesoderm. The key difference between a diploblastic and a triploblastic animal is a diploblastic animal produces two germ layers. Just look the first picture. You have an ectoderm and you have an endoderm. But where is the mesoderm? Instead of a mesoderm, there is a portion called mesoglia, which will act as a subordinate of the mesoderm. Instead of mesoderm, in case of diploblastic animals, we have mesoglia. That's just a difference between a triploblastic and diploblastic. Tri means three. You can easily understand. In case of diplo, di is used for showing two. So triploblastic means they have three germ layer. In case of diplo, there's only two. So outer and just inner. That's enough. So the fourth one is the classification on the basis of coelom. In case of the classification on the basis of coeloms, the coelom is the fluid-filled body cavity present between the alimentary canal and the body wall. Alimentary canal means through the center, and the outer wall, that is the outer surface of the skin. So coelom is a fluid-filled body cavity between the alimentary canal and the body wall. The true coelom has mesodermal in origin. They are being produced from mesoderm. So what is a mesoderm? We have just studied that mesoderm is something that is found in between the ecto and the endoderm. The perionic cavity present in the abdomen and similar spaces around the organism such as the lungs, heart, parts of a coelom. Coelom differs in the structure and formation process. So just take an example. You can simply understand it from the example itself. Take the case of a flatworm. If a flatworms, they are acoelomate. Usually, a word which having starting with a, acoelomate, that means they are absent in coelom. Asymmetric, they have also a is there.
In case of acoelomate,
so that means they lack coelom. See, there is no space between alimentary canal and the body wall, so they doesn't have a coelom. In case of the pseudocoelom, the word pseudo means fake, false. This is the meaning of the word pseudo. Pseudocoeloms, such as example is the roundworm, they have a coelom, but they are not a real coelom. They are just false. In case of arthropods, that is the higher animals, they have the true coelom. So this is just a classification on the basis of coeloms. So there are three types. So we can study in detail what is acoelomate, pseudocoelomate and eucoelomate. So in case of an acoelomate, the coelom is absent. The plastocoel is completely occupied with mesoderm. There is only spongocoel and coelentron present. That is, there is no coelom, right? There is no coelom. So in that case, only spongocoel and coelentron is present. The examples are Porifera, Coelenterata, flatworms. That is an example of Platyhelminthes have such character. And only spongocoel and coelentron are present. So the next one is pseudocoelomate. That is, they doesn't have a true coelom. They lack true coelom. The blastocoel is partly filled with mesodermal cells. Mesodermal cells are being inside the blastocoel. The body cavity is being lined with mesoderm only towards the body wall, and mesoderm is not present towards the gut. The only example we can say is roundworm. This is an example for Escalmindus. So next one is a eucoelomid. Usually U means true. They have true something. It's what we call a U. So animals that having true coelom. Coelom is lined with a mesoderm on both of the sides towards the body wall and towards the gut. The blastocele present in the gastrula gets completely replaced with the true coelom. The body organs are suspended in the coelom by mesenteries. For example, this from phylum Annelida to Chordata. In this case, in case of euchordates, they have a true coelom where the organs are being found in the coelom suspended. Now the next classification is on segmentation. So I hope you all seen earthworms. You may have seen some type of a cut in between the two segments, or the segmentation is a division of some animal or the plant body plants in the series of repetitive segmentation. For example, in case of the earthworm, the body shows a pattern called metameric segmentation and the phenomenon is called metamerism. In case of earthworm, whatever they have in the first segment, it will be found in the last until the last segment. That's what we call a segmentation. Now, classification of notochord. This is the very important topic in this, because we are primarily classifying an animal account with the notochord. If there are notochord present, if their notochord is absent, if they are found in the primitive stage, we are describing everything on the basis of notochord. So notochord is a flexible and supportive role found in the protochordates, formed a material similar to cartilage. Cartilage is something, you have a nose, you have ears. You can flexibly bend these ears, right? It can be moved up, down. You can bend the ears because that's made of cartilage. So notochord are some flexible rod-shaped structure found in protochordates, and the material is similar to cartilage. That is, they are flexible enough. It's not bones. The notochord serves as the base of an axial skeleton. The axial skeleton is a part of skeletal system that consists of the bones from head to the trunk of the vertebrate. The axial skeleton is from the head region, that is from the skull to the trunk of the vertebrate. So it consists of 80 bones and is composed in 6 pairs in the skull, that is the 22 bones. The ossicles of the middle ear, the thyroid bones, the rib cage, the sternum, the vertebral column. So the notochord serves as a basis in the axial skeleton and induces the overlaying ectoderm to differentiate the neuroectoderm to form the neural plate. They are derived from mesoderm, that is, they are formed from the middle portion. Except the jaw vertebrates, notochord are replaced by the backbone. There are some jawless fishes, jawless vertebrates. Excepting for them, all the notochord are being replaced by a backbone.
This is all for today, and this is the first part of animal kingdom. So in the next video, we will be talking about the diverse animal kingdom detailedly. So the book I am referring to is NCERT textbook. For in case of degree level students, it's Animal Diversity Part 1 and Part 2 I am referring to you. So I hope you understand the portion. I hope you all like the video. And don't forget to like, share and subscribe to my channel. So let's see in the next video. Till then, it's Anamika signing off. Bye.